striking examples of organisation within the animal kingdom. They achieve massive efficiency by dividing their resources into reproductive and non-reproductive efforts. The leader of this organisation is the queen, who lays all the eggs within the hive. Once they are laid, the eggs develop into larvae and eventually pupae. Most bees enter the working stage of their lives as hive bees. As they get older, they take on more complex roles, such as foraging to bring back pollen for the colony. The future queen is chosen at just 3 days old, and their diet is immediately changed from nectar to royal jelly. Queens are significantly larger than a normal bee. They live longer, and they are the only female in the hive with fully developed ovaries. Queens enter the reproductive stage of their lives just 16 days after they are laid. This begins by making their one and only trip out of the hive, where they mate with many drones. Once they return, they remain in the hive for the rest of their life, laying up to 2,000 eggs a day. Despite extensive research, it is still unknown if queens are chosen by a social factor or a predetermined genetic basis. However, in 2011, Masaki Kamakura made an important discovery when he determined how royal jelly induces the worker-to-queen change. After being stored for 30 days, Kamakura observed that the effects of royal jelly had completely degraded. He isolated different elements of royal jelly and tested their properties, and found that the protein 57KDA matched this behaviour. This protein was designated as royal lactin, the factor in royal jelly that induces queen differentiation. However, bees are not a typical model organism, and Kamakura needed access to a wider knowledge bank to finish his research. Surprisingly, he found that royal jelly induces the same morphological changes on Drosophila as the bees, making them the perfect match to continue his testing on. To uncover the signalling pathway of royal lactin, Kamakura used both RNA interference and the Gal4 system. RNA interference is used to manipulate gene expression by degrading target mRNA strands before they can be translated into proteins. A complementary double-stranded copy of the target gene is made and inserted into the cell. The enzyme dicer breaks the strand into segments called small interfering RNA, or siRNA strands. The siRNA strands are then transported by the complex RISC to the target mRNA. The two strands bind and the mRNA is degraded. The Gal4 system is another method of isolating specific genes to determine the role of the proteins they express. First, the Gal4 gene is placed under the control of an enhancer and a specific gene. An upstream activation sequence, or UAS, which is under the control of the Gal4 gene product, is placed into the genome of another fly.